Welcome to Part B of my lecture on bioenergetics and mitochondrial functioning. In Part A, we focused on bioenergetics and defining all of these parameters. Now in Part B, we will focus on the mitochondria itself. And we will begin by talking about the chemiosmotic hypothesis and how it generates ATP. But then we'll spend the bulk of our time on mitochondrial diseases and how we can understand them better in the context of our knowledge about bioenergetics. Let's start with the big picture of energy metabolism that we've been discussing. Carbohydrates, proteins, and fats all break down eventually to acetyl-CoA to provide this key intermediate in the matrix of the mitochondria where they generate NADH units. That NADH passes its electron to the electron transport chain, by which process a gradient is generated in the intermitochondrial space as indicated by the protons. And these protons and this gradient drives the synthesis of ATP, which occurs back on the inside of the matrix and generates these important energy units for the biosynthesis of macromolecules. First, let's introduce some terminology. Mitochondrial DNA is a circular DNA genome that's found in all mitochondria. It is separate and distinct from nuclear or chromosomal DNA. And the mitochondrial DNA is always maternally inherited. And we will come back to talk about this later in the lecture. The second term is reduction potential, and that represents the voltage difference between the reactants and products in a redox reaction, which is related to the free energy of the reaction. And it underlies understanding the electron transfer process. The third term is that of an uncoupler. It's a molecule that can shuttle protons across a normally impermeable membrane, such as the inner mitochondrial membrane, and remove the gradient that is set up in it. The classic molecule is dinitrophenol, which you can see has an equilibrium between its protonated and unprotonated form. And it is the protonated form that can diffuse across the inner mitochondrial membrane. So if one adds dinitrophenol to the mitochondria, it will diffuse across the membrane and destroy the gradient. It uncouples the gradient. The free energy change for a half of a O2 molecule to pick up a H plus and a hydride, an H minus, or two H pluses and two electrons is 0.82 volts. Similarly, for an NAD plus to pick up a hydride or an H plus and two electrons to make NADH is minus 0.32 volts. So the net reaction in electron transport is that an NADH plus a half of an O2 is converted into water, and that's a free energy change of 1.14 volts. The delta G0 for this is minus 52.6 kilocalories per mole. The delta G0 required to make a high-energy phosphate bond, that is, the conversion of ADP plus a phosphate to make ATP, is about 7.3 kilocalories per mole. Now, since NADH yields through the electron transport chain the equivalent of 3 ATPs from the gradient generated, that would be a total of 21.9 kilocalories per mole that is then contained in the ATP so generated. Comparing that to the reaction on the previous slide suggests that the efficiency of the production of ATP from the energy generated is 42%. And this is typical for biological reactions of about a 40% yield of energy or efficiency. I've also noted the similar conversion with kilojoules if you're used to that nomenclature. Now we come to the link of the electron transport chain to ATP synthesis. The previous two slides dealt with the energy numbers involved. Now let's talk about mechanism. And to explain that mechanism, one invokes the chemiosmotic hypothesis. This was first proposed by Peter Mitchell, and like the LDL receptor hypothesis that I introduced in a previous lecture, it is very difficult to prove a hypothesis correct. One makes the hypothesis and then carries out various experiments that, if they work as hypothesized, would be consistent with the hypothesis. Peter Mitchell made this hypothesis and then invoked various evidence from several experiments in support of it. And I'd like to just summarize those. First, if you measure the pH on the outside of a mitochondria versus inside, there is a pH difference, and it is lower outside, consistent with a gradient of protons that are sent outside the mitochondria. Second, if you take a mitochondria and you impose a gradient by external sources, you can make ATP. Third, if one takes reconstituted bacterial rhodopsin plus the enzyme ATP synthase, which I'll introduce in a moment, and shine UV light on it, you can make ATP with the light being a source of energy picked up by the rhodopsin. Fourth, if you take submitochondrial particles made by various processes like sonication of mitochondria, in which they turn inside out, the synthesis of ATP is vectorially oriented in the reverse direction. Fifth, one always needs an oriented membrane to achieve oxidative phosphorylation, and if you do anything to compromise that, it doesn't happen. And finally, I introduced uncouplers like dinitrophenol in an earlier slide, and when one adds those two membranes, then ATP synthesis comes to a halt. So those are 6 pieces of evidence that support the Mitchell chemiosmotic hypothesis for how the link occurs between electron transport, the main subject of Part A of this lecture, and ATP synthase, which we're going to turn to next. We now come to the ATP synthase itself, the enzyme responsible for the synthesis of ATP. It is, after all, an enzyme, but a very complex enzyme with multiple subunits, and it's often referred to as complex 5 at the end of the electron transport chain. Like the other complexes, it too is embedded in the membrane, and it has an orientation in which the outside faces the intermitochondrial space and the inside faces the matrix of the mitochondria. The enzyme is composed of multiple subunits. There are 6 subunits comprising the F0 portion, which is embedded in the membrane. There is a stalk which leads to a set of 3 beta subunits and 3 alpha subunits, which are arranged as depicted and known as the F1 portion. This enzyme is oriented in the membrane, as I indicated, and the proton gradient from the outside can be viewed as passing up and through that stalk region, driving ATP synthesis from ADP on the inside. Let's now look at the whole process. The electron transport chain generates a gradient from the matrix to the inner membrane space. That is, protons are exported to that space, and therefore it has a high proton content or a lower pH than the matrix. That gradient is dissipated when the protons flow back inside through the complex 5, or the ATP synthesis enzyme, to convert ADP to ATP. In summary, we should think about this as a completely integrated process of electron transport driving the synthesis of ATP, which provides in the matrix the energy units to drive biosynthesis. We can now turn to mitochondrial diseases, taking advantage of the background about the electron transport chain and ATP synthase. This is important because, as I mentioned earlier, mitochondria have their own DNA. Pictured here is the mitochondrial genome, and while it only codes for a small number of proteins, they're very much involved in these processes. Indicated on this slide are the coding portions for ribosomal RNA genes and transfer RNA genes, and importantly, certain proteins that are components of complex 1, complex 5, which is ATP synthase, complex 4, and complex 3. And indicated on the inner circle are certain mutations which lead to certain syndromes, which we will point out later. As I discussed in Part A of this lecture, the complexes of electron transport are composed of multiple subunits containing cytochromes. Interestingly, only a small percent of the proteins in these complexes are coded for in mitochondrial DNA, and the remainder are transported in from the nucleus, generated proteins. So the small number of genes in the mitochondria includes 2 ribosomal RNAs and 22 transferred RNA genes. But complex 1, which consists of 46 coded proteins, has only 7 derived from mitochondrial DNA. Atp Synthase, which has 18 different proteins, contains only 2 coded by the mitochondrial DNA. And complex 4, known as cytochrome oxidase, has 3 of 13, and complex 3 has 1 of 11, whereas complex 2 codes for no genes in the mitochondria. So, in summary, mitochondrial DNA is maternally inherited. There are multiple copies of the DNA in mitochondria, and so one could have mutations in some copies and not in others, which makes for a very complicated genetic inheritance situation. Of the 100 or so electron transport genes that need to be coded for, only 13 derive from mitochondrial DNA. And in total, mitochondria code for 37 genes, as indicated here, including especially a number of transfer RNA genes. And we already know over 200 point mutations in the mitochondrial DNA that have associations with diseases. Now let's look at 3 typical mitochondrial diseases. The first is Leber's hereditary optic neuropathy, and it, like the others we'll look at, is an uncommon disease, rarely found. Leber's disease is picked up by ophthalmologists typically, who observe a swelling of the retinal nerve fiber layer. The symptoms that occur and bring the child to an ophthalmologist are progressive loss of vision, sometimes accompanied by headaches. This is a complex 1 defect of one particular protein in that enormous complex. And like the other mitochondrial diseases, there are no cures, but there are some experimental agents that will help treat the disease. Another example is Malaise Disease, which is again uncommon, but it's the single mitochondrial disease that is picked up by pediatricians. Its symptoms include a neurologic ATP loss resulting in stroke-like episodes, sensory failure, lactic acidosis, and usually they don't see a ragged red fiber that is otherwise typically present. Again, this is a genetic disease, And here it's a defect in mitochondrial DNA, usually a transfer RNA leucine defect. There is a treatment for it, but it is a treatment and not a cure. Now, recall at my very first slide in my first part of this lecture, I introduced this case of a 13-year-old girl who had a 2-year history of worsening symptoms, a variety of symptoms as listed here, none of which alone sort of points to a specific disease. And this is a typical presentation of mitochondrial diseases where there are several features that are not quite normal, but not necessarily a dominant single feature that points to a specific mitochondrial disease. The third example is MRF, and this is a neurologic disease that presents with epilepsy, ataxia, hearing loss, and one can see ragged red fibers on the muscle tissue. Again, a mitochondrial disease, and again, in the mitochondrial DNA of a transfer RNA, in this case, lysine. And again, there are some supportive treatments, but not curative treatments. In summary, there are numerous mitochondrial diseases. We talked about Leon, which has a main symptom of blindness. It's a complex 1 defect, malaise, strokes, and lactic acidosis, which is a transferred RNA, specifically leucine defect. MRF presents as epilepsy and myopathy, and is a transferred RNA for lysine deficiency. There's another common one, Karen-Sayre disease, which has a number of symptoms, and the defects can vary. But none of these are curable, but some can be managed to some extent with existing therapies. What's important to recognize is there are a small number of mitochondrial genes, and they are responsible for some mitochondrial disease defects. But over 90% of the pediatric presentations of mitochondrial diseases turn out to be due to nuclear-coded genes, which produce the bulk of the proteins involved in the electron transport chain and ATP synthesis, and often they are involved in the assembly of these complex proteins. In summary, mitochondrial diseases are very rare. They often involve multiple organs. So when one sees a variety of sort of apparently unrelated symptoms involving 3 or more organs, perhaps a common disease like epilepsy, plus something out of place someplace else, then one should look further into a mitochondrial disease origin. But we should keep in mind that mitochondrial diseases are uncommon, they are progressive, and they're usually picked up sometime in childhood. So let's come back to this picture. Energy metabolism is at the center of life, particularly if we think about the liver that has so much oxidative phosphorylation occurring to produce ATP to make important biomolecules. And the heart of the oxidative phosphorylation process and the generation of ATPs starts in the matrix of the mitochondria, generating an electron gradient and driving through this gradient the synthesis of ATP, which then can be used as energy equivalents for biosynthesis. We should also keep in mind that the coding of these electron transport chain proteins, including the ATP synthase, is heavily nuclear, but some small and definitive number of proteins are coded by mitochondrial DNA, which makes for a very complex genetics. Finally, this lecture has been packed full of detailed information, so I want to conclude the lecture by reviewing what it is one really needs to know and get out of this lecture. So first of all, with regard to protein gradients, Part A, one should understand the principles involved in gradients, the major carrier molecules, how the energy flows in the mitochondria from where things are located and where they move to. The features and function of the cytochromes in heme are central to this, and there are numerous clinical applications from understanding the electron transport chain. With regard to Part B on ATP synthesis and mitochondrial disease, it's important to again understand the principles of energy capture. Know the efficiency of biological reactions in capturing the energy in the form of ATP, but you don't have to memorize the delta G values in those equations, but have the sense that about 40% of the intrinsic energy is captured in the form of ATP to drive the important biosynthesis of our central molecules of metabolism. Try to understand the chemiosmotic theory, which is an explanation for how ATP synthase functions, and it is based on its interesting and important structure. And understand the supporting evidence, the 6 items I named, that support and help to explain what the chemiosmotic theory of energy capture is composed of. So, in understanding the big picture of energy metabolism, one needs to know what happens where, in the membrane, in the matrix, outside. And, of course, the major clinical applications and when one should suspect mitochondrial disease.